Greetings. I'm Robert Lee Kilpatrick, the chair of the member-led Forum for Health and Medicine, and I'm also on the board of governors of the Commonwealth Club World Affairs in San Francisco, California. And I'd like to thank all of you in our audience in YouTube land joining us today for this really very interesting program. I'm delighted to host Dr. Paul Alan Cox, who is the executive director of the Brain Chemistry Labs in Jackson Hole, Wyoming. And he's come to the Commonwealth Club since 1903. We have been producing programs of interest to people across the spectrum of society. And of course, there's nothing more interesting than Alzheimer's, ALS, and Parkinson's disease, because many of our friends and families suffer from these things. So Dr. Cox is here today to talk about his work as an ethnobotanist, working with indigenous peoples around the world to understand what he refers to as tangle diseases. And so, Dr. Cox, thank you. I'd love to hear more about this topic. Welcome.
Well, thanks. And great to be at the Commonwealth Club. And many thanks to you, Robert, and all the good people behind the scenes that are making this possible.
Can I say one thing? Those of you in the audience, if you have questions, you can put them on the chat box on YouTube, and we'll have a look and do our best to present your questions. Sorry to interrupt. Thank you.
So if it's okay, Robert, why don't I launch into a presentation for maybe 20, 25 minutes and then turn the time back to you? Would that be okay?
Thank you very much, Paul.
Okay, great. So this is where our laboratory is in Jackson Hole, Wyoming, where the Brain Chemistry Labs were a not-for-profit organization. And my real goal here, Robert, is at the end of our conversation that we can bring new hope to people, because there's some very exciting things happening with these tangled diseases. Fortune, think of this, they came out and they interviewed me. I didn't get my picture, but I got my silhouette on the cover. Here we are. Very interested in the different approach that we have to Alzheimer's disease in Wyoming. Sanjay Gupta, who's a medical correspondent for CNN, became very interested in our work as well, wrote about us in his book, and brought a film crew out and made a short documentary. So I'll show that to you right now, if that's okay. So let's see here.
When we started looking into dementia and Alzheimer's, I didn't expect to be coming here.
There's the Grand Tetons. You're looking at the Grand Teton National Park.
Nestled in these majestic Wyoming mountains is Dr. Paul Cox and his small nonprofit called Brain Chemistry Labs.
We're a little bit off the mainstream here.
I first sat down with Paul and talked about his unorthodox approach 5 years ago in 2019.
I want my scientists to have ideas that they can pursue that are different than the current ideas. It looks almost like they're invading the amygdala. Basically what we're arguing is amyloid is not the cause of Alzheimer's, it's a symptom. So is tau. These are the symptoms. This is like trying to look at what caused a train wreck by investigating the wreckage. We'd rather go way up the tracks and look at the switches, and that's what we're doing genetically in our studies, is try to find out what are the very basic roots of protein misfolding, and if so, is there a way to stop that?
It's an audacious theory that has been years in the making, coming not from a medical doctor, but from an ethnobotanist.
Ethnobotanist. What exactly is that?
I'm trained to spend my time with indigenous people and see how they use plants for medicine. And if we start looking at the pattern of illness and wellness among indigenous peoples as a way of discovering clues to major illnesses, this can sometimes pay off.
His novel theory on Alzheimer's disease has its roots on the tiny island of Guam in the Pacific. It started with a mystery surrounding a bizarre mix of ALS and Alzheimer's symptoms.
In Guam. What did you see there?
At its peak, a quarter of the people in these villages were dying of this disease. Every single family I visited with had some family member that died from the disease.
In Guam, the Chamorro villagers were seeing grave neurodegenerative symptoms at 50 to 100 times greater than anywhere else in the world. Dr. Cox told the world about it in 2003.
We discovered that they were eating these flying foxes, and that was their most salient food item. We went to every family we'd interviewed. There's a toxin in your food, that these flying foxes, that the cycad seeds have this toxin. And I didn't know what they'd say, and they said, "Thank you. We didn't know what was making us sick. Now maybe we can save our children."
Cox's disease detective work unfolded like this. Cyanobacteria, which is often described as blue-green algae, contains toxins such as BMAA. In Guam, BMAA was accumulating in shallow pools of water, which was then leaching into the cycad trees. Those trees were then yielding seeds high in BMAA, which were eaten by flying fox bats. And then humans were consuming those large bats because they're considered a local delicacy.
When I first started reading about this, Paul, I thought, okay, well, this situation in Guam, that's got to be a pretty unusual situation.
Yes.
But the idea that some version of this could be happening in other parts of the world, is that likely?
Yes, and what terrified us is when we realized that cyanobacteria might be the culprit. It was like staring into the abyss because we realized you could be exposed anywhere.
What are you saying then? If people are thinking about Alzheimer's, thinking about these neurodegenerative diseases, the likelihood that cyanobacteria, something that looks like this, could be the culprit or at least part of the culprit, how likely is that?
I'd say it's a risk factor. It's one path. There's probably many risk factors for Alzheimer's. And at this point, our hypothesis is that exposure to these cyanobacteria increases your risk of certain neurodegenerative illnesses.
To figure out just how big a risk factor, Cox's team took cyanobacteria and replicated those toxic effects of the BMAA in vervet monkeys.
You had one group of animals who got placebo, basically the normal diet, another group that got the toxin, BMAA, and a third group that got the toxin and L-serine, this amino acid.
And that L-serine reduced the density of neuropathology by up to 85%, depending on the brain region. So that suddenly we thought, wow, this potentially should be considered as a possible way of treating Alzheimer's.
A simple amino acid known as L-serine.
If you also gave the L-serine
with the toxin, boom,
it seemed to be protective.
It's protective. It was these data that we took to the FDA that led into our advanced clinical trial.
How do we get L-serine? Is it something that we normally get in our diet?
Yeah, L-serine is rich in our diet. I mean,
what kinds of food?
Eggs are rich. Anything involving soy tofu, edamame, sweet potatoes, turkey, pork. You know, there's a lot of sources. But what amazes me is the average American gets from all of their dietary sources about 3 grams per day of L-serine. These people in Ogimi are living to be 100 years old. They get 4 times what we get.
I spent time with the people he's talking about as part of my Chasing Life CNN original series.
Here in Japan's tropical south, on the island of Okinawa, it's what's called a blue zone, which means it's home to some of the longest living people in the world.
Yeah, this one. Do you still eat a lot of these today?
Now, Dr. Paul Cox is taking that Okinawan diet high in L-serine and putting it to the test here in the United States at Houston Methodist.
Essentially, the purpose of the study is to find if the L-serine is able to help these patients slow down or even stop the progression of Alzheimer's disease.
Using these jelly beans, researchers are giving 30 grams of L-serine a day for 6 months. It's a placebo-controlled trial of 100 patients with mild cognitive impairment.
They have to remember words.
Led by neurologist Dr. Gustavo Roman, changes are measured through cognitive tests, PET scans, and blood biomarkers.
This is always a hard question, but if it were your relative, would you tell them to take L-serine?
We're confident it's safe now, but we want to make sure it's efficacious before we suggest anybody try it. We're going to know for sure in a short time if L-serine can treat mild cognitive impairment, which is a precursor to Alzheimer's. If it's a treatment, it also might be a preventative, so stay tuned.
Now, halfway through the trial, results are expected in 2025.
Robert, did you have any comments or questions on that clip?
Actually, what I wanted you to clarify, are you saying that diet, in this case, containing more L-serine, would be a risk factor or a cause of Alzheimer's?
No, actually, if our hypothesis is correct, L-serine consumption reduces your risk of Alzheimer's.
Okay.
This little village that they have so much L-serine in their diet through the seaweeds and tofu they eat, the edamame, they don't have any incidence of Alzheimer's or ALS or Parkinson's disease. So it reduces your risk.
Okay.
But what we are saying is that cyanobacteria and the toxins they produce are a risk factor and can increase your risk if you're exposed.
You know, that was my only question at that point. The video is fabulous. Maybe you can carry on.
Thank you.
Thank you.
Okay. There we go. So we started a not-for-profit organization in Jackson with a singular mission of improving patient outcomes. We get highest ratings from the charity Navigator, charity things. We have 6 senior scientists that anchor our lab in Jackson Hole. Then we have a consortium of 50 scientists around the world who work with us on a weekly and sometimes a daily basis. They come over, they visit, we do experiments in their laboratories. So what we really have is sort of like a virtual pharmaceutical firm. We have focused not on symptoms, but on stopping protein misfolding. And that's maybe why Fortune said we're a radically different theory, because we believe that since these diseases are caused by misfolded proteins, if we can stop those misfolds, we're confident that we can come up with new ways of preventing and treating ALS, Alzheimer's, and Parkinson's. Here's Dr. Milo Lin, who was a Miller Fellow at University of California, Berkeley, holding a slinky. If you're like me and had them when you're little, they get tangled pretty quickly. Turns out the slinkies actually have this alpha helical conformation, which is similar to neuroproteins. And this is from an Alzheimer's patient, a mat of misfolded tau. So in Alzheimer's disease, there's 2 proteins or protein fragments that misfold, tau and beta amyloid 42. But in ALS, it's different proteins that misfold, TDP43, FUS, SOD1, a few others, and in Parkinson's, alpha-synuclein. And so our thought was if we could stop the whole process of protein misfolding, not only could we treat Alzheimer's or ALS, but we could treat half a dozen other neurodegenerative diseases that are characterized by protein misfolding. I recruited a real star group to go with me to Guam. Dr. Susan Murch, now endowed professor at the University of British Columbia in amino acid chemistry, Professor Sandra Bannock, who was from Berkeley and then Cal State, me, and we had some neurologists and physicians. We studied intensively 2 villages over a series of expeditions where the rate of death from these sort of diseases was 25% of all adults. We got very interested in cycads, which they were using to make flour. I knew as a botanist that cycads produce aerial roots that are up in the photosynthetic realm. They harbor cyanobacteria. Here I've cut with my penknife one of these root tips so you can see the cyanobacterial areas. And they produce this unusual neurotoxin called BMAA, which is an unusual amino acid. The people take the kernel of the seeds, They make the flour for tortillas, for dumplings. They also eat animals that eat the cycad seeds, including flying foxes and pigs, deer, other things that eat the seeds. So the hypothesis we published in Proceedings of the National Academy of Sciences was that these cyanobacteria produce this unusual neurotoxin. It accumulates in the trees, particularly in the seeds, through various items in the traditional diet. It's ingested by the people and kills them. We've been monitoring other sources of BMAA. You don't have to go to Guam and eat a traditional diet to be exposed. Cyanobacterial blooms that appear like blue-green scums on the water also can produce the toxin. So we've been very carefully monitoring the water coming out of Lake Okeechobee in Florida, down the St. Lucie River to Stewart on the east coast, and down the Caloosahatchee to Fort Charlotte on the west coast, exposing lots of residents unwittingly to this neurotoxin. Very pleased that yesterday the Corps of Engineers has announced they're going to stop these releases, and they've adopted a new management plan that will hold that to a minimum. So I'm very grateful we've been working with the Corps on this. We found early on with our colleagues in Australia, Dr. Rachel Dunlop and Dr. Ken Rogers, that this neurotoxin inserts itself in proteins in place of one of the normal 20 amino acids, serine. We know that that's common in soybeans, seaweeds. So we wanted to see if it could block insertion into proteins. So in human neuronal cell culture, we actually found if we increased the amount of serine in the culture medium, we could stop protein misfolding. So we wanted to see if we could replicate this in an animal model, went to St. Kitts in Guam, where we worked in vervets. Here's part of our team. It's a very nice facility. There's 40,000 vervets that were introduced into St. Kitts years ago. There's no natural predators. They actually outnumber the people now. But there's a nice facility here, a full-time veterinarian care. So we began an experiment where we'd offer the vervets fruit with a substance embedded after they had their dinner. The ones that had placebo had absolutely no neuropathology, but the ones that had the toxin had these very characteristic neuropathology, which was identical to what we'd seen among the Chamorro villagers. But then we found that if we gave them toxin but added serine to their diet, that we could block this neuropathology by 85%. So here we have tombstone neurons filled with misfolded tau protein, neuropil threads made of the tau, and then all sorts of neurons in various states of degeneration. We also found that we could block the protein misfolding associated with ALS. So here's rice flour, placebo. Here's the spine when the animal's exposed to BMAA. We get FUS, ubiquitin, TDP43, protein misfolds. So this really, we reproduced the neuropathology of the villagers. Most of them had an ALS type presentation. Some had Parkinson's symptoms, others had Alzheimer's symptoms, and we were able to totally replicate all observed neuropathology in the villagers in this experiment. I wanted to find then a sort of a human analog, because we'd been in these two villages in Guam that have the highest rate. Was there a place that had none at all? This led me to Ogimi Village on the northern top of Okinawa, known as Longevity Village in Japanese, where the diseases are unknown. Here's a simple lunch that one of the villagers prepared, rich in different types of seaweeds. They ate 25 different species of seaweed. They eat tofu. They have a lot of L-serine in their diet. In fact, they get 4 times what we get in the United States. This is a normal villager, age 98. She's not extraordinary, she's just normal. Watch this for a second. She has no neurological deficits. I asked her if she could touch her knees, and she went over and put her palms flat on the puppy mat. Watch this. I can't do that. Wow. Well, maybe we have acrobats here, so I asked her if she could bend
後ろは
backwards.
大丈夫後ろ
So she bends backwards. That wasn't such a good idea, she said. But she's 98, of course. Then I want to see this neuron that goes from your spine down to your ankle, your legs movement.
膝こうやっ
Asked
て
her to
上げ
lift
る
her legs here. Look at this. I mean,
こっちは
it's
難
just
しい
terrific. Remarkable people. Again,
音を聞きな
she's normal. She's not extraordinary. So we took the data from the villagers, from the animal studies, from our neuronal cell cultures, the FDA. They, within 4 days, gave us a license to begin a phase 1 trial of L-serine. It's a safety trial we conducted in Phoenix and also in San Francisco. We gave the patients different doses. This is a slope. The people at the low end of the slope are progressing very rapidly in ALS. We found at 30 grams per day, we could get a significant reduction in ALS progression. We then began a phase 2, which got interrupted during the pandemic, but we'd finished 28 patients. Here's the normal patients from 430 patients from the placebo arms of failed drug trials. Here's our patients. We were able to slow their progression by 32%. And we're looking for funds now to repeat this study with a full complement of 50. We also began a phase 2 trial for mild cognitive impairment. That's a precursor to Alzheimer's disease. People with it can still hold a job and follow a recipe or drive, but they're having glitches. It's led by Dr. Gustavo Roman, who's one of the world's top Alzheimer's doctors. He's head of the NANCE National Alzheimer's Center. Dr. Edwin Vega is a neurologist, as our clinical coordinator. For 100 patients, we're doing PET scans before and after treatment. They're treated with L-serine or a placebo at Houston Methodist Medical Center. It's a double-blind randomized trial, but we're optimistic, and we'll have the data completed certainly by this time next year. So stay tuned. We've also been very interested in speeding diagnosis. This is our senior scientist, Dr. Sandra Bannock, Dr. Rachel Dunlop from Australia we hired. We've now developed a fingerprint for microRNA that is up to 98% accurate in just from a single blood draw in distinguishing ALS patients. And we're hopeful that we can actually identify patients before they develop any symptoms, thereby really speeding diagnosis, because many patients have to wait a year and a half or more to get a diagnosis. And we're having a paper come out in the next week or two in Oxford's Brain Communications, disclosing this data. We're trying to find a diagnostic firm that can make this available to neurologists around the world. We've also invented a new drug for advanced Parkinson's after levodopa stops working, and to protect cognition. Here's the team that of our scientists that worked in developing this. So we're pretty excited. Based on our 7 expeditions to Ogimi, we've come up with the decision that people can actually create their own blue zone. One thing everybody can do is get moving, get more exercise. 30 minutes a day of a brisk walk really reduces the risk of Alzheimer's. Most of the women in Ogimi garden, and we find that gardening may be part of the solution. Stay engaged. They get up in the mornings, they sing, they dance, they have little Peter Rabbit games I can't even keep up with. And this is part of building your cognitive reserve. Oliver Sacks, who is my tutor in neurology, felt very strongly that music is empowering, ennobling, and helpful. His patients, some who could not speak, could sing. Some who could not walk could dance. And then diet, the Mediterranean diet has been repeatedly shown to reduce risk of Alzheimer's disease. This is a diet based on fresh fruits and vegetables with a lot of antioxidants, olive oil, very little red meat, a few fish. So there's different things we can do to reduce the risk of Alzheimer's disease. And furthermore, I'm certain that it's not inevitable, because I've got this whole village in Okinawa have never heard of Alzheimer's disease. They were really horrified when I told them it was an age-related form of dementia in the United States. So our next steps going forward, and I'll conclude with this, is we're trying very hard to make these new microRNA blood diagnostic tests, move them into a diagnostic of a commercial company so that they can be made available to neurologists to help speed neurologists' diagnosis. We are completing the phase 2 mild cognitive impairment trial in Houston. And once that's completed, we'll break the blind and see how the patients are doing. We have raised half of the $1.5 million cost for a new Parkinson's drug that we've developed for a trial. And we've raised $400,000 of the needed amount for a phase 2 ALS trial for L-serine. So we're very excited. It's my real hope, Robert, that this brings hope to the people here. You can learn more about our work at brainchemistrylabs.org. Again, we're a not-for-profit organization. I'm very grateful for the many people in the Bay Area and elsewhere who are assisting us achieve this mission. So I think with that, Robert, I'll stop sharing and turn the time back to you.
Thank you, Paul. Well, there's so much to talk about, so much to think about. I love that concept of making your own Blue Zone. Of course, I've seen the Blue Zone series on Netflix, but I've never heard anyone say, well, you can make your own Blue Zone, because the idea that's always told to us is that you have to go somewhere to an island with a tiny population to benefit. So that's a very interesting idea that you suggest. And as a keen gardener myself, living on coastal California here, where we garden year round, sorry to tell you that Wyoming gets a little colder. So you mentioned gardening, exercise, staying engaged, music, diet. All of that sounds like lifestyle.
That's correct. And, there's things that can increase our risk. I should have mentioned that these animals we studied all had a double dose of the APOE4 gene, and the human beings is a risk factor for Alzheimer's disease. So, unless you're clever in choosing your parents, you might not do that, but you can do lifestyle things. And I'm really quite convinced, and I think there's broad consensus in the neurological community. In fact, a study just came out Monday from London, again, showing the power of changing diet and reducing Alzheimer's. Exercise, one thing I didn't mention is learning a new language or even attempting to learn a new language also statistically reduces risk of Alzheimer's disease. So why not build your own Blue Zone by, even if you're on a small island called Manhattan or why not build your own by doing some lifestyle changes?
So, we began with you talking about being trained as an ethnobotanist and then going, to places and working with indigenous cultures. If I think about what's called the standard American diet, SAD, what people actually eat in this country, and the huge obesity rate, and I think about my childhood when we didn't have computers and we didn't have cell phones, and my mother used to say, you get to watch television for half an hour a day, and that's it. So, here we are on a computer screen for an hour. So it strikes me that one of the differences between then and now in the United States is people are more sedentary. They're in cars a lot. I mean, I was raised in a small town and we just walked everywhere. People are on screens all the time. They're on their phones. So I just wonder, as an ethnobotanist, when you go to Okinawa or you go to Guam or some of these places, is their lifestyle like our modern lifestyle of all this sitting and watching, or are they more active than we are?
Oh, they're much more active. In Okinawa and Ogimi, they get up in the morning, they play these sanshins, these 3-string banjos.
Yeah.
They sing, they dance, they move. They're active in the garden. So I think for whatever reason, the trajectory of our industrialized societies is concerning because all of us, let's face it, Robert, spend too much time on our screens when we could be out walking, exercising, visiting with the neighbors, staying socially engaged The Commonwealth Club's a good example. People that are engaged and intellectually engaged build what we call a cognitive reserve. It's sort of like filling water in the reservoir, which again can reduce Alzheimer's. And then diet is a big, I don't think this is contested. In fact, the new study from London showed that if you eat ultra processed foods all the time, that you really do have an increased likelihood of Alzheimer's disease. So being more careful on our diet, being more active, these are common sense things, but it turns out statistically that they're very powerful in reducing risk of Alzheimer's disease.
So my experience in the Chinese diaspora or the Indian diaspora is that both of these large and ancient cultures have a concept of food as medicine.
That goes through most indigenous cultures, that they do not divide food and medicine as separate categories. For example, the Hausa in Nigeria changed their diet during malaria season and started eating anti-malarial plants. So that it seems that in this aspect, we're unusual as a culture in that we have these as separate categories.
It seems in the Western world that food is not seen as medicine, it's seen as entertainment, it's seen as pleasure, it's seen as profit, all of these things. And so it seems like no matter what you say, and it's a compelling argument, it is a big lift for many Americans to imagine not going to drive-through restaurants, not, actually eating more like our grand or great grandparents ate in this country. We used to eat fresh. People had gardens when I was a kid. Most people had a little garden, and that seems to be a thing of the past. So I just wonder if, when you're talking about what you see in Okinawa and Guam and other places compared to contemporary United States, I'm beginning with the large concepts before we drill down. Is it a cultural thing in part that you're talking
Yeah,
about?
it really is. And one thing that's been very striking for me is to work with indigenous people that then become westernized and move I have a colleague, a wonderful ethnobotanist in the University of Arizona in Desert Botanical Garden, Gary Nabin, who's
Mhm.
extensively studied indigenous Native Americans. And I mean, type 2 diabetes is exploding once you go on what he calls the Circle K diet, the not to pick a particular chain, but just to say that
Yeah.
eating ultra processed food and foods that are like white rice or white bread that have a very high glycemic index really seems to be associated with loss of health among indigenous people. One thing I have to say, I'm sort of excited. I know about you, Robert, but have you ever tried pickleball? I mean, that's something I gotta believe. Getting people out playing this stuff is going to be a way of get people moving again.
Yeah.
Getting people moving, walking, eating locally, growing their own produce. These seem to be good trends to me.
Well, again, not to push I'm a native-born Californian, but certainly here along the coast, it never freezes, and we don't have great heat. So it's easy to be very active outdoors here, perhaps less so in the winter in Wyoming.
Well, we probably have more extreme skiers per square
Oh,
meter in Jackson, Wyoming than anywhere in the world other than maybe Chamonix in France. And Wyoming is playtime in the winter. We're skiing, we're sledding, we're cross-country skiing, ice skating, walking. So I hate to say it, Robert, but maybe you've been a little spoiled as a native Californian because there's lots of ways to stay active in the winter.
maybe I've just confessed indirectly that I'm not a skier. Um,
This is a
So-
curable malady. Come up to Jackson.
Thank you. Thanks for
guidance
the
and lessons.
invitation. I'll hold you to that. So
Good.
Paul, I'm trying to get clear in my mind the general statements around what you saw in Guam and what you saw in Okinawa. Are you saying that the richness of L-serine in the diet in Okinawa is a major contributor to the absence or the lack of Alzheimer's, and in Guam it's related to the cyanobacteria? I'm not quite sure how those two stories fit together.
Right. Well, let's talk about it in the Dickens sense, A Tale of Two Villages. Okay. In Guam, people were pretty good until after World War II. They were given semi-automatic weapons. They then had the ability to really start decimating the flying fox population.
Okay.
These big flying foxes eat the cycad seeds. If you eat a flying fox, you're getting a massive dose of neurotoxin. They extincted one species in Guam, put the other on the endangered species list.
Mhm.
I've made 2 interviews with Chamorros that went to hunt the remaining flying foxes, which is in a single colony at Anderson Air Force Base, which is a
Okay.
nuclear weapons base. They took their weapons, went across the fences under the wing of the B-2 bomber, shot a flying fox, and exited, which either suggests to me that these people have an insatiable appetite for flying fox flesh, or our much vaunted Homeland Security still is not up to snuff. But they were getting such massive doses of BMAA from cycad seed flour from these flying foxes that 25% of the people died.
Okay.
I mean, it was huge. Now let's move to the other village in Okinawa. The people are moving there. They are eating a highly enriched serine diet, and they avoid food items that I guess they wouldn't know this, but would have an enriched amount of BMAA, such as shellfish. They won't eat shellfish, and they're eating this amazing sort of seaweed-based diet. Yet the climates are pretty similar, so I think it's a great natural experiment to sort of show that you can live healthy in those circumstances if you have the right diet, the right exercise, the right approach to lifestyle.
So I've got a question from our online audience, and I think it's a good question, and it may not be the easiest one to answer, but I'll let you have it. So, how much of Alzheimer's is hereditary versus environmental? And by environmental, they are talking about poor diet, low exercise, exposure to chemicals and frying pans, chemicals in food. So is there any way for us to generally understand the hereditary or genetic component and then the environmental component? Because the L-serine could be both, couldn't it? Couldn't it be environmental and genetic, or is it just environmental?
Well, let's talk about that for a second.
Okay.
We know that in Alzheimer's disease, that about 10% of the cases are familial, about 90% are sporadic. What's
Mhm.
interesting, we have about the same percentages in ALS. About 10% of cases in ALS are genetic, and half of those patients have an identified mutation. The other 90% are sporadic, and similar in Parkinson's. And I think Parkinson's may be the better case um, because there it's been really carefully tied to exposures to certain sort of pesticides, including rotenone, Agent Orange, and now glyphosate. So I think there's broad consensus that these progressive neurodegenerative diseases are a gene environment interaction. Problem is that almost all the research has been going on the genetic side, and it's been wonderful research, but I think the environment has not been carefully focused. In the last New York Times Magazine, there was a very interesting article in New Brunswick, a mysterious case of people coming with an unusual neurodegenerative illness. I asked Professor Murch, why don't you get some of the lobsters out of that place? And boom, they're chock full of this toxin.
Mm.
I think there's many different risk factors. I just want to emphasize that. Many different risk factors that can lead to Alzheimer's. In the case of the Chamorro people of Guam, I think it's clear that it's BMAA, and we replicated that in laboratory animals. But what's fascinating to me is in our clinical trials, it's clear that L-serine seems to be protective against all forms. And so this is an amino acid that maybe has been overlooked. We produce a little bit of it in our body. We get about 3 grams from our diet. But, you know, these people in leukemia are getting 4 times that.
Yeah.
And but I also want to not discount their lifestyle,
Yeah.
because they're active, they're engaged, they're intellectually, you know, it's a combination of risk reduction factors, I think, that are functioning in Okinawa.
So early on in your presentation, you mentioned that mild cognitive impairment is a precursor to Alzheimer's. How do you define mild cognitive impairment? Because once in a while, occasionally, I can't remember a name, but I had that when I was 5 years old too. So I'm not sure it's age related. What does that mean, mild cognitive
Well, a
impairment?
person with mild cognitive impairment has notable glitches in their cognition, and they start feeling something's wrong with me.
Okay.
It can really only be diagnosed by a really competent neurologist,
Mhm.
but it turns out that about 70% of people with mild cognitive impairment go ahead to develop Alzheimer's disease.
Wow.
And
Okay.
so we've admitted 100 people. I think we're up to enrolling 72, 73 now in Houston with this diagnosis. Half are getting jelly beans with serine, half are getting placebos, and we're carefully measuring them. And we want to see if the ones that are getting serine have a reduced amount of mild cognitive impairment.
Okay. Um, one thing I'd like to clarify, and I know this is about the science, and so since most of us on this call are not, practicing scientists, perhaps, so please be kind and careful with us in your answer. This notion of tangle diseases, to be honest with you, Paul, I have not heard that term before, and yet when I looked at your pictures, it is a perfect description, and the slinky was a good way of presenting it. Can you talk a little more about what you believe causes the tangles? I know you talked about protein misfolding, the tau and the beta amyloid 42 for Alzheimer's, the TDP4S for ALS, and the alpha syncline
Synuclein.
for Parkinson. So what do you think is the contributor to the tangling?
Well, let me just say this, that progressive neurodegenerative diseases, which include the three we've talked about, Alzheimer's, ALS, Parkinson's, and some others such as chronic traumatic encephalopathy that football players get, or supranuclear palsy, Lewy body dementia, they're progressive. They always get worse. They never get better. And this is very different. Some people have an essential tremor that just stays the same for 20 years. A
Okay.
person with Parkinson's disease is progressing. And neuropathologically, we can see that they start accumulating these protein tangles. So it's a process that, first of all, protein tangles begin, then they get sticky. So they stick together. We call that oligomerization. And then once they're sticky, they start pulling in like a little neurological black hole, all sorts of junk, and they become very toxic to our neurons. So what are the things that can cause protein misfolding? Well, obviously, some environmental exposures can, certain genetic proclivities can. We probably have a clearance mechanism that clears out these tangles as they occur, because misfolded proteins are very dangerous for neurons. But perhaps as we get older, those clearance mechanisms lose their efficacy. And I'd say the strongest correlate with progressive neurodegeneration, like Alzheimer's disease or ALS or Parkinson's, is age.
Right.
One thing we published in a while back in Proceedings of the National Academy of Sciences was based on our work in Guam and work with some other great folks at NIH, Ralph Garudo being primary. We found if villagers leave those two villages and they move to California, Southern California, they can be gone 10 or 15 years and then they get the disease, and it's the Guam disease.
Yeah.
So 10 or 15 years after any possible environmental exposure, they get the disease. So we said, based on that, we believe there's a latency period in these diseases. We said up to 20 years in Alzheimer's. I don't
Right.
think the world was ready for that sort of announcement from some cowboys in Wyoming. But now I think most neurologists agree that there's a long latency period where you're asymptomatic and you're just building up this sort of overburden of protein tangles. And actually, that gives me a lot of hope, because if we can identify people before they develop symptoms, we could begin treatment. And
Yeah.
any treatment's going to work better in a disease at the earliest stages at the later stages. So that's why we're so focused on these microRNA blood diagnostics that we've developed.
So do you have any idea of the relationship between L-serine and the Tangle diseases? I mean, you understand that there's some kind of a correlation, but you don't have an idea of mechanism. Is that right?
We have some ideas about mechanism. For example, in our neuroproteins, 92% of the phosphorylation sites, this is a site where the protein is charged so it can fold properly, those are serine residues. So if there's a misincorporation event then of the neurotoxin there, it's going to really result in misfolding. We also know now that serine alerts these little sentinels in our endoplasmic reticulum looking for misfolded proteins.
Yeah.
It also prevents creation of ROS, reactive oxidation species. So just like when I was a kid, they said, Wonder Bread builds strong bodies 12 ways. That's what the bread. It looks like serine has a number of mechanisms. The thing that I find so attractive about it is twofold. First of all, in our clinical trials so far, it seems to be highly effective. Certainly in animals, if you have a monkey that's going having some problems, let us know. We know how to cure it. The question is, will that make it across to the clinic? And the jury's out on that one. We have to see when these data come back. The other thing that excites me about serine is it's so inexpensive. You
Yeah.
can buy it by the wheelbarrow full. We're estimating that drug costs for an Alzheimer's patient would be about $32 a month, $30 per month. This contrasts with some of the recent amyloid busting drugs that were released and subsequently withdrawn that were costing $150,000 per patient per year. So a real trick is finding a pharma company that can construct a value proposition from marketing such an inexpensive but powerful dietary amino acid.
Well, yeah, there might be other ways to do it, but that's another conversation. One of the things that really excited me about your presentation, I'd like you to say a bit more, is this blood. Is it miRNA test?
Well, if you think of DNA as a keyboard,
Yeah,
microRNA are the fingers that play the keyboard. They turn the genes on and off.
right.
And we found now a group of 8 microRNA that uniquely define an ALS patient. And we had a clinical trial where, at diagnosis, we're taking blood. So these are, and then the CDC's really helped us out. They've come on board and given us lots of samples. I think we're up to sample size of about 600 patients. And it's very accurate in identifying ALS. Furthermore, this is way upstream. And let me sort of explain what I mean. By the time somebody's having symptoms, it's like a train that's already been derailed. By the time these protein clusters and tangles occur and are replete within the brain tissue, if the focus is trying to destroy those, that's sort of like trying to fix a train wreck by reassembling the wreckage. In our team, we go way up the tracks. We want to see where those switches, genetic switches are. So we're at the earliest stages. And our diagnostic test is not designed to detect protein misfolds. We're actually detecting changes in microRNA. And so we're very excited about this. And stay tuned. I think there'll be a little bit of a splash in the news in a week or two on
So
this.
are you looking at biomarkers, or what are you actually measuring?
Well, yes, microRNA is a biomarker. And if you have, they're small RNA sequences. They're about 22 amino acids long. And if you have these particular 8, we can say, hey, this is probably an Alzheimer's patient. A neurologist should carefully look at that. If they don't have those, then we say no, and we have a pattern for healthy control. So it's a very accurate test in terms of biomarkers for diagnosis. What we don't know is if it's good for prognosis. In other words, can we see if a drug is working by giving it to you and see if it changes the microRNA? We don't know the answer to that yet, but we very much would like to know. It'd be fantastic if true.
Well, I think you said, but unless I wrote it down incorrectly, this blood test, its purpose is to identify ALS patients before clinical symptoms can be seen. Is that
We
accurate?
would love to do that. And my dream would be that in the United States, that once somebody gets into the age where ALS is a significant risk, say your 50s or 60s, colonoscopy age, that a single blood draw is taken from your arm, it goes off. Most people don't hear about it. Very
Yeah.
few contacts say, we'd like you to meet with a neurologist to go further on this.
So what's your development plan for that test?
Right now, we're in discussions with a couple of diagnostic firms. One just rang me here during this talk because we're a not-for-profit. We're good at discovery. We're not set up to commercialize things. So we'd like to transfer this technology to a diagnostic firm. And ditto for L-serine. We've got great patent counsel at Pillsbury Winthrop. We'd love to transfer those patents to a pharma firm that can then take it into phase 3 trials and FDA approval.
So, one of the historical features of the Commonwealth Club is that we don't sell products. That's
I hope
that's
I haven't here,
no, but I'm going to give you an opportunity to talk about something. And I'm setting the stage. So we don't sell products. However, you said that you've raised half of 1.5 million for a Parkinson trial and 1.1 million for phase 2 ALS trial. So I'm going to give you an opportunity to talk a little bit more about those two trials, a little bit about what they look like and why some of our audience might be interested in getting involved in this.
Well, I want to be very respectful of the Commonwealth Club rules. And
Yeah.
so cut my mic off if you think I'm going south here.
Okay.
Our phase 2 trial for mild cognitive impairment at Houston is costing about $2.4 million. Clinical trials are very expensive because you have to pay for the physicians and neurologists and everything. We already have a good idea the phase 2 ALS trial will work based on our analysis of the first 28 patients. And we think we can run a very fast trial on that one for 1.5 million, and we have $400,000 in hand for that one.
Okay.
We also developed a new drug for advanced Parkinson's. We've raised about 750,000, a little bit more towards the, which is half of the cost of that trial. Do I want to sell this to the Commonwealth Club members? No, but I would say there aren't a lot of folks walking around that can say, hey, by the way, I sponsored a clinical trial and here's a drug. I mean, just think, if you were to donate, Richard, and we succeed in the drug, maybe we could prevail on the pharma company to call it Kilpatrickin, and I mean, who knows, but the idea in a very small amount of money, these are fractions of the cost of what pharma companies spend on clinical trials. We can discover potentially a new drug. That's exciting to us and potentially exciting to some people who are philanthropists there.
Yeah. Well, I don't need my name on a drug, but no, the reason why I'm asking you this question is because these tangle diseases are awful. I mean, they are devastating to individuals and to families, and to communities. And so we all know people. I know people. A dear friend from high school is in the final stages of ALS, and it's just incredible tragedy. So there are people out here, perhaps in this audience, who might like to in some way support this or be a part of this. And so I think that you have a really good concept, and so I don't think we're not breaking any rules. I just want to say I think good science should be supported, and so I encourage everyone to consider that.
Thank you. And Robert, I'd like to say that just looking at you, you are already doing the things that reduce your risk. Can you scoot sideways? I think I saw 2 saxophones there behind you.
Yes, you saw 3 actually.
Can you scoot away so I can see them? They're right behind you.
Yep.
Scoot your chair sideways. And then, okay, there we go. There's the saxophone. Then I'd like you to go over and grab one of those green books. So that's a classic series. Grab one and read me what the title is, please.
Okay. Well, this one is appropriate.
There we go. Hippocrates. And then you remember the Commonwealth Club. I'm assuming that you're still active in California and walking around even in the winter. So you're already
I live on an organic farm and grow my own food.
bingo. See, so you're doing it. You are your
I'm
own.
141 years old and you'd never know it.
I wouldn't put you a day past 139, so that's
Yeah.
great.
No, I think, I mean, this is a very fascinating, I think this notion of the ethnobotany approach versus a pure science in the lab, somehow you've managed to kind of marry the real world examples of people living their lives in different cultures, and at the same time using cutting edge science and techniques to be able to decipher what is happening in the real world. That's an exciting story.
It's really fun. And I'd love to take you to this village. In fact, take all of our listeners. I mean, it's incredible to be with these people. Maybe it's a placebo effect, but when I'm eating their diet and watching, hanging out with them, I feel a little bit more spry and a little bit more, step. And so the idea that each of us could create our own blue zone, which I think you're doing very effectively, is pretty exciting. I got to have dinner last night, 2 nights ago, with an 83-year-old woman. I said, "What have you learned since you were 70?" She said, I've learned is the power of poetry."
Oh,
And she's
wow.
reading poetry. She loves Tennyson. And we are talking about that. And she in poetry courses declaimed and read aloud. And so the power of music is something Oliver Sacks believed deeply in for his patients. I don't know how if there's been double-blinded studies on that, but it seems to me that you're a great example, Robert, of creating your own blue zone.
Well, thank you. I'm definitely going to use that concept, Paul. And I wanted to say that while I don't ski, I do fly fish. So maybe next spring or summer, if I come out to your area and go fishing, I could pop in and visit you and see your lab.
The cutthroat trout are singing to you. And I have some good friends who are supporters of the lab, have their own fly fishing ranch. So please come up. And I should tell people we welcome visitors at the lab. Love to show you what we're going on. It looks like a log cabin with a carved science bear on the front. This thing's wearing a lab coat and goggles and holding a flask. And I've got some of the smartest people in the world working for me in that lab. And I guess my message really is one of hope. With
That's
Dr. Sanjay Gupta, we were talking, he and I both believe, think about this for a second, that the last Alzheimer's patient is living today. What we mean by that is that Alzheimer's will be off the radar screen. It will be destroyed within the lifetime of somebody that's alive today. And when I was a little boy, I used to walk with my mom into the hospital lobby. There'd be all these iron lungs, people with polio. Jonas Salk came up with a new way of thinking about polio. He was my boyhood hero. Wouldn't it be great if something as simple as tweaking our diet, getting exercising, listening to music, learning a language, if those things actually could change us so that this does not become an issue anymore? The last Alzheimer's patient is alive today.
That's wonderful. Now, I want to be super respectful of our online audience. And so I have two questions. I'd like you to try and keep your answers short because we have about, 5 minutes and then I'm going to close it up. But I want them to feel that, I listen to them. So
Good.
here's the first one. Both of them, I think, are related to issues that they personally are experiencing, and that's why I want to mention them. The first one is someone who says, I have mild cognitive impairment, and I've read that lecanemab, a monoclonal antibody, might be beneficial. What can you tell me about that?
Yes, it's been marketed by Eisai. It's being prescribed for people with mild cognitive impairment. It's been approved in the United States, not approved in Japan where it originates, which is interesting. And I'm not sure of the status in Europe. So really my advice is always listen to the doctors very carefully. They're going to do their best for you with the standard of care. I'm personally concerned about drugs that attack amyloid in the brain. There's, some side effects, brain and bleeds and sort of things like that that happen to people. But at this point, um, I think listening to the neurologists and following their advice is a good way to go forward.
Okay. Thank you for that. Now, this is our final question, and then I think you can close with a couple of positive statements. The last question is, going back to the 8 biomarkers, if you do detect them, then what happens? Where does a patient go if they're diagnosed with those biomarkers?
Well, at this point, it's a research project,
Yeah.
so we're not directly dealing with patients in our lab. But the idea is when it's commercialized, everybody would get this test. Most people, since fortunately ALS is a rare disease, they would not show those biomarkers. The few that do, say 2.5 per 100,000 people, would be referred to a neurologist. And then the neurologist can have those data. We're also developing biomarkers for other neurodegenerative diseases. So we think this could really help neurologists much more rapidly make a diagnosis. Furthermore, currently it costs about $5,000 in mobile visits to a neurologist to get a diagnosis. Right now, for a couple of $100, this test could be done. So our real hope is that we can speed very rapidly diagnosis so people can know where they stand.
So my final comment before your final comment is and it's kind of a question. Would it be fair to say, Paul, that one of your main theses is pay attention to your health?
Well, absolutely. And that's one thing that's been so exciting to me in working with these people who are over 100 years old out there chopping wood and dancing. I mean, it's changed my whole view of aging. And I guess my final comment is, as an ethnobotanist, I need to say that Alzheimer's is not inevitable because there's a whole village I've been living at, working with, where they have never even heard of the disease.
Right.
So the changes that we're talking about, get moving, learn a new language, listen to music, exercise, build your cognitive reserve. These are things that require some changes, but they're not onerous. They're pleasant, will improve your quality of life. And L-serine, jury's out. We're going to have to wait till we get the results back from our clinical trial, but we're guardedly optimistic, and we'll let everybody know when we find out whether it works or not.
So thank you, Dr. Paul Allen Cox, the Executive Director of the Brain Chemistry Lab in Jackson Hole, Wyoming, for an incredibly interesting and fast-moving, can't believe the hour's gone, program. I'd like to thank our audience for coming in and listening to this. I would strongly encourage you to go to their website. And what's the website again?
brainchemistrylabs.org.
There we are.
And if they want to see the CNN clip, they can just click on it there, go to YouTube, put my name and Alzheimer's in, and it'll punch up.
Okay. I also want to encourage everybody in the audience who is not a member yet of the Commonwealth Club of California to consider joining for a mere $10 a month. And so for $10 a month, you get all sorts of benefits. It's a wonderful organization founded in 1903. It is the nation's largest and oldest public affairs forum. And now we've merged with the World Affairs Council. Go to www.commonwealthclub.org to see our amazing slate of programs for the next weeks. You can join and do come back. I hope to see you again. And Dr. Paul, it's been a pleasure to talk to you. Thank you for
Honored to meet you, Robert. And I just hope next time I can hear you play that saxophone. So
I'll bring one with me to Wyoming.
there we go.
Thank you.
Thanks, everybody.
Bye now.
Bye.